So Mendel noticed that when he looked at how things grew, when he looked at characters, when he made crosses. Now remember, he was a monk. He was an Austrian monk. In a time before television, before radio, when people just, your pastime was reading. And remember that the reading material wasn't that, you didn't have a lot of reading material. You had to actually go to a library who owned books, unless you were rich. And if you were rich, you had your own library. That's what made you rich. So having a lot of books, having a big library was almost a class status symbol. So Mendel, the reason I mention that is because if you can imagine someone taking and growing peas in a garden and then growing these peas and these pea plants and then crossing them. And you have to think about what it takes to do that. So you're going to grow these teas. Some of them are tall, right? And some of them are short. Some of the plants grow tall, some grow short. Some have yellow peas in them. So when you open up the pea pod, you've seen peas, right? They look something like this. You open them up, and inside there could be yellow peas, or they could be smooth, or they could be green peas, and they could be, rough. So he noticed that these differences, and he started crossing them. Now imagine what you have to do to cross them. You have this flower, horrible drawing, but this flower, and you have this little place where all the pollen sits, right? And I don't know if you knew this, but a flower is the sexual organ for the plant, right? And here is a gamete for the flower, right? There's the flower's gamete. We call it pollen. It's the male equivalent to the male sperm in humans, right? And there's another gamete, usually in a flower at the bottom. Now, in the case of the pea plants, that not all plants do this the same. What's the key to sex for plants? Plants that do sex, what is the key here? Yeah.
The bees take the pollen?
Right, the bees do spread the pollen. The bees will come in here to get nectar, and the pollen will stick to the hairs on the bee, the little tiny hair-like structures that stick out. And when the bee goes from one flower to the next, the pollen gets shaken off. But the key to sex in a plant is that this gamete, this pollen, has to get to the egg. Now, in the pea plants that Mendel studied, the pollen would fall. A wind would blow and the pollen would fall. So they can mix their gametes among themselves in their own body. So you could get a plant having sex with itself and producing more of itself. Does that make sense? The wind could also blow some of that pollen out, right? And by the way, this is why people are allergic to certain pollens, because you breathe it in. And of course, it's going to find another plant, another flower somewhere else. And if that happens, then you have a cross between the two plants. Does that make sense? So what Mendel had to do is he had to take this really tiny pair of scissors and a little scopula, and he had to go in and cut the stamen, the thing with the pollen on it, and transfer the pollen from one flower to the next. He had to make sure he did that carefully, because if he didn't do it carefully, then he couldn't tell any results, right? So imagine doing that with thousands of plants over many, many years. It takes a lot of patience, right? But he was a gardener, he's a monk, he didn't have a lot of other things to do, he didn't have children, didn't have a wife. So he had the time to do it, is the point. And so that's the process. And when he noticed something about this, though, once he started crossing them, he said, look, whether it's green color, yellow color, right, the color of the pea, or if it's wrinkled or smooth, what I notice is that they're not dependent on each other. When you get yellow, they're not always smooth. When you get green, they're not always wrinkled. You can get wrinkled in pea. And you know what else? After he collected a lot of this data, right? A lot of this data, he noticed that first thing he needed to do is find what he called this. He called them this, true breeding. What does true breeding mean? Yeah, they always have this certain trait. You have a trait that's always there. So if it's tall and you cross it with another tall, it always equals a tall, right? If it's yellow and you cross it with a yellow, it's always going to equal a yellow, right? It's always, these are the traits, right? That he's always seeing this. He's like, look, no matter what I do, I cross a yellow with a yellow and I get yellow. Now we know true breeding. We've known about true breeding since the days of the Egyptians. Like we crossbred the Egyptians. Have you ever seen the drawings of cats on their pyramids? They bred cats, right, for thousands of years. And dogs is another breed. Well, how do you think we do this? When we call a dog a pit bull, or we call a dog a greyhound, what do we mean? Yeah.
You're talking about probably the different trait and the type of dog that they are?
The traits and the types of dog they are is correct. But what do we mean by that? We have a trait. If something looks like a pit bull, is it a pit bull? How do we know? We have a whole American Kennel Society. We have a British Kennel Society. What do? They certify a dog as a breed. So what is a breed? Yeah. They have a certain set of characteristics, you're right. But what about those characteristics? When I mix them with another dog that has those characteristics, what do I get? A dog with those characteristics. And we call it what? What do we call that? True what?
Breeding.
True breeding. So that's why we call it a breed. That's why we call it a breed. When we say that a dog breed like a Cocker Spaniel or a Cockapoo or whatever it is, we say it's a breed. We mean that when you take this dog, this Cockapoo, and you mate it with another Cockapoo, you're going to get what? A Cockapoo. I mean, that's their characteristics you're going to get every time you mate them. We call that true breeding. There are problems with this, and we'll talk about that as we move along. Yeah?
When you were talking about how Mendel moved the blood from one to another with like the scissors and all that stuff,
Mm-hmm.
couldn't like something affect like the outcome, like the air around or something?
Absolutely. He had to take a lot of things into consideration, didn't he? He did it in an abbey. He probably was doing it in a courtyard, so there's not as much wind as in a field, let's say. He was probably there was a lot of things that came into this. It was very lucky. Mendel was very lucky or blessed or whatever, right? He's very, very lucky. Because had he picked another plant, this wouldn't have worked. Had he picked some other traits, it wouldn't have worked. It turns out that this particular plant, the characteristics that he looked at, they were dominant or recessive. Had he looked at some other plants with some other characteristics, it wouldn't have worked out that way. Because it could have been, like you've already read, theoretically, or you better read before too soon if you haven't. There are other ways of inheriting other than dominant recessive. There is, codominance, right? Incomplete dominance, multi-gene inheritance. So there are things, like you could not take two people of the same skin tone and mate them and guarantee that their child will have the same skin tone. It just won't happen. There's no way to guarantee it. They always have a variation of skin tones. Why? Because there's more than one gene involved. There's no dominant and recessive. It's not that as easy as that. Now, as I said, albinoism is that easy because there's a master gene. If you have this broken master gene, then you're an albino, and that's it, because you're not making any melanin. But the different shades of melanin, you have, no matter what the population, you always have variation in skin tone. The reason you have variation in skin tone, no matter what the population, is that you have these 8 genes. Now, if you have a population that's been isolated for a fairly long time, the population could tend to lean towards being lighter or darker. And if you have natural selection going on, like in Africa, for instance, or in Australia, where lighter skin is a real detriment, then you're probably going to get less and less lighter skin over time. And then in the north, where dark skin is a detriment, you get less and less darker skin people, and over time you get more and more lighter skin tone people, because the darker skin people don't survive in the north. Oh, by the way, the only reason those of us who have darker skin survive in the north, although I'm kind of in between myself, between the lightest and the darkest rather. Darker skinned people survive today in the north because of vitamins. Otherwise, there would be some real problems with darker skinned people, especially in the winter months. Does that make sense? Because do you know that you make this vitamin called vitamin D? Do you know how that vitamin's made? From the sun hitting your skin. So it's not just the sun. You're not just the sun going through the glass. You have to actually be outside. So it's a problem today. Most people don't get enough vitamin D. Why? Because most people don't spend time outside. So one of the things, and it's hard to get it in dietary foods. You don't get a lot of it in dietary foods. So today we have milk. We have vitamin D supplemented milk. We have vitamins. These are the things that encourage us to be healthy up in the north. Now, the darker your skin, the harder it is to get this vitamin D from the sun. It works well if you're in a strong sun, like in Africa or Australia or even Southern Europe, where you get a lot of sun all the time. It's fine. But when you're in the north, you get less and less sunlight. Now you got issues. Question? Yeah, there are many genes, and when we're talking about skin tone, and I don't want to get off topic too much here because we really need to get into the math of this. But just to keep, I think it's worth the time to think about what we're talking about here. So you have gene. There's a gene. Let's call it A. There's a gene B. Let's think about what this means. All right. Let me minimize this for a second and. So let's think about what this means. You have gene A, you have gene B, you have gene C, and you have gene D. These are genes, right? These genes make proteins, and these proteins might impact a gene, it might make another impact other protein. So there's all these biochemical pathways that we've studied. You know what I'm talking about. We're changing, even though you don't understand the specifics. I don't understand the specifics. It doesn't matter for the intents and purposes of this conversation. So these are all making proteins, and they're all impacting each other. Some might be impacting each other. Some might change this enzyme, might change that enzyme, which changes this enzyme, which makes this protein, which makes this protein. And you get a certain amount of melanin. It gives you, that melanin gives you a certain shade of skin tone. And I can't really do shades here for whatever reason. Can I do shades? No, I guess not. All right, well, you get all kinds of shades of skin tone from this. This might be, I don't know. Let's say there's a 1 from a 1 to a 10. 10 being the darkest and 1 being the lightest. And there's all kinds of stuff in between, right? And again, we're going to do a lab on this. There's a really great genetics lab that looks at multi-gene inheritance, a paper lab. I think you'll be interested to see that. So you have all kinds of skin tones in between. Does that make sense? Yes? No? Okay. So maybe this particular combination gives you a skin tone of a 1. But then you change something. You get, what if there's actually 8? I put A, but actually A2, A3, B1, B2, C2, C3, D2. How many is that? 1, 2, 3, 4, 5, 6, 7. That's too many. There's something like 8 genes involved here. All right, 2, 4, 5, 6, 7, 8. So there's something like 8 genes involved here. Okay, so see how complicated this is. And they can influence each other. They can influence all kinds of things. You get a melanin 1. Now, all you got to do is change this one. Instead of getting a B1 allele, you get a B, lowercase b1 allele. Now you get a skin tone of 2. So maybe you don't get a B1, maybe you get a B here, but then you get over here you get a lowercase d1. Now you get a skin tone of 3. So it gets kind of crazy, doesn't it? There's all kinds of different combinations that give you all kinds of different shades of skin tone, which explains why, no matter where you are, we all have different skin tones. There's not, it's not white or black, it's shades. It's not shades of gray, if you will. Right? So, but there is a master skin, there is a master gene. Without this gene leads to all, this is the master gene for all melanin. If you have capital X, no problem. You can make all these different other proteins and they can end up giving you a skin tone anywhere from 1 to 10. Okay? But if you get instead this lowercase, right? Now nothing works. It doesn't, the melanin, the master gene doesn't make any melanin at all. So you get not a 1 to 10, what do you get? A 0. That's an albino. Someone that can't make any of the melanin, any of the proteins, because the reactant that each of these needs, the precursor that each of these needs, is not made. So it can't make, so none of these can be made. Does that make sense? Okay, so that is multi-gene inheritance. Here, anytime you get not one or the other, listen to what I'm saying. If you're dealing with one or the other, you're probably dealing with dominant or recessive, right? But when you get a bunch of different values, right? Shade color, eye color, height. Height is not tall or short, is it? We're not all 8 foot or 3 foot. It's not a binary choice, is it? Is it? No, it's not. There's various levels, there's various gradations, shades of gray. Right? So when you're dealing with that kind of inheritance, you're talking about probably multi-gene inheritance. Does that make sense? Yes or no? Please help me. Yeah, okay. If it doesn't, you need to say something, because that's how you end up not coming to me after the test and saying, I didn't get it. I'm like, well, why didn't you say something when we were talking about it? Huh? Yeah, go ahead.
I was looking at this thing about dog breeds, and I found out that golden retrievers, if they're purebred, they're more likely to get cancer due to the lack of genetic variation, and bulldogs. Due to their bone structure, it's nearly impossible for them to give birth without dying, so they have to get a C-section.
I want to talk about this, and I will talk about this, and it's absolutely right. And here's the thing. When we breed, this is the problem with race, discussing race in humans as well, by the way. When we as humans breed dogs, we're looking at certain characteristics, right? In a golden retriever, it's a webbed foot because and the ability to go in, and they're soft mouth dogs, so they don't bite down hard unless they're trying to defend themselves, right? They'll just lightly grab things. We want that if you want them to go retrieve your duck, right? You don't want to send a pit bull to grab your duck because if he brings it back, he's going to bring back a broken duck, right? Because that dog is going to crush it. Because that's what pit bulls are. Pit bulls and terriers were bred to kill. They were bred to kill rats. They were called rat dogs. They're actually very, very safe dog breeds, by the way. And people don't like them, mastiffs either, because they're powerful dogs. And the problem with pit bulls and mastiffs is that human beings train them to bite humans. They were never bred to bite humans. They were bred to go after rats. That's why you'll see a terrier react quickly and they shake like this. That was something we bred into them. We liked that because when they grabbed that rat and they shook it, they broke the rat's back. And you don't want the rat. It's serious business because rats can get big, right? And did you know that rats can get pretty big, almost like cats? Yeah, some rats, small cats, but still, some rats are big. And if you're gonna send a dog to go get a rat, you need to make sure it kills that rat quick because otherwise that rat's gonna bite your dog. And you don't want that rat biting your dog because it could have disease or hurt your dog, take its eye out or whatever. So you're breeding a warrior dog. Does that make sense? You need to have that instinct of grabbing, shaking, killing, moving to the next one. And so they have to be high energy. They have to be able to shake really quickly when they grab. And that's what you get with terriers, all terriers. And a bulldog is called an American Staffordshire Terrier, but there's a British Staffordshire Terrier as well. And those are what we call pit bulls, although pit bulls are generally not true breeding because we have other dogs mixed into them. So in any case, the point being that when we look at 2 or 3 characteristics, right, shaking, strength, jaw strength, or whatever, we don't look at everything else. When we looked at bulldogs, for instance, we wanted big heads and small bodies because we thought that looked really cool. Right? And everybody here thinks so because when you look at a pit bull, everybody loves a pit bull. Why? Because they have giant faces, right? And strong shoulders, and they're pretty skinny bodies, really, right? You ever see them? So that's the breed. That's the breed that the people that like those dogs, that's what they go for. Well, with the problem with the English bulldog is that they kept breeding for that characteristic, but they didn't think about the consequences. So now in the English bulldog, the head's too big, so it can't come out of the mom. The mom would die. Everybody would die if they can't. Most English bulldogs have to be born via C-section. So it's a real problem. In fact, some breeders have started breeding smaller-headed English bulldogs so that they could help fix that problem. The issue is that nature would never have allowed that to happen. The first mother that had a baby whose head was too big, that mother would have died. The babies would never have been born, and those genes would never enter the population. Does that make sense? So nature would never have allowed that to happen. Humans have bred this trait, and then we've also fixed it surgically, but now we have an issue. And by the way, there's other problems as well. When we have these big heads, they have smaller noses, so they have problems breathing. And they have to have surgery, often thousands of dollars worth of surgery, to fix the problems that we've bred into them. So we're looking at 2 or 3 traits, but we don't think about the other things. Also, when you're inbreeding, which is what we're doing to make these, right? We're not moving genes around. We're breeding brother with sister, cousin with cousin. That's what we're doing. Do you understand that? When we're talking about making a true breed, we have to take this brother and mate it with this sister to have a baby. What that does is if there's any recessive genes in there that cause disease, they're going to show up. And some of the diseases are hip dysplasia, joint problems, cancer. These are all consequences of inbreeding. The healthiest population are mutts, but people don't want mutts because they don't have the look that they want. Yeah, question?
So how do they actually
You don't pick a gene. You pick a what? You're not picking a gene when you're breeding. What do you pick? A trait.
No, but like how they make it so like the dog has like a little bit smaller. Or something.
You pick the smallest dog, and you made it with what? Another smallest dog. You breed the two smallest dogs until you get, and then their children are probably going to be smaller-ish. And you take the two smallest dogs of that generation, and you make them. You take the two smallest of that generation, you make them until you get tiny dogs. Does that make sense? You're just sticking to the traits. This is what Mendel did. Of course, he did it with peas, which I think is a little more, a little safer, humane. I don't know if it's humane. I don't like that term humane because humane means human-like, human characters. And humans don't, we're not that nice. So, but humane, ideally humane. Like, what is our human ideal, right? All right, crossing true breeding plants. So we take true breeding plants. So I know that this is always tall, and this is always yellow. Are we clear on this? And the independent assortment, he said, look, I don't know what this thing is. Because remember, he didn't know what genes were, right? He didn't know what DNA was. He didn't know what proteins were. He had no idea. He says, look, there's something in this plant. And I know that at least in part it's the pollen, and I know it has something to do with the pollen and the egg. Something inside the pollen and the egg that gets sent to the next generation. I don't know what that stuff is. It's something. He called it a thing, a packet, a particle, something, right? I don't know what it is. He didn't know what it was, but he said that something was getting sent, obviously. Because when I have two tall plants, I move the pollen around, I can make a short plant. I can make a short plant, or I can make a plant tall just by mixing the right particles together. Does that make sense? He didn't know what they were, but today we know it's DNA. We know they're chromosomes. We know that those chromosomes are in the sperm, in the egg, in the pollen in this case, in the egg. Yes? Yes? Okay. So he didn't know, but as he's crossing them, we know today that those things that caused that were alleles, right? We know they're alleles. And we know because of his numbers, he said, look, I'm going to call these things dominant, and I'll call the other one recessive. I don't know what it is, but I'm going to call them dominant, recessive. And he said, for the yellow ones, I'm going to say the Y, big Y is that he's using letters as variables because he doesn't know what they are, right? And by the way, this notation is stuck. We use it today, don't we? This is over 100 years ago, and he did this, and we're still using it because it works. So capital Y is dominant over lowercase y. How do I know that? Because I cross them. Every time I take a yellow, true breeding yellow, and mix it with a true breeding green, what do I get? Every time I take a true breeding yellow, people, which means what? If I say it's true breeding, that means they're homozygous. That's what true breeding means. You're homozygous. True breeding yellow, and I mix it with a true breeding green, right? Homozygous. What do I always get? I always get a yellow. Today we know that means it's a heterozygous, right? So if we did the Punnett square that you practice with the guinea pigs, by the way, I hope you did that guinea pigs thing because that's due. Does everybody realize that? That guinea pig, you guys, this class didn't get it. Someone pass those out. I'm sorry, the other classes got it already. My bad. So this is just a practice. This is a practice at guinea pig. So that'll be due soon. All right. You want me to make it today? Okay. All right. Maybe whatever you like, whatever's best for you. If you want a deadline, I'll set the deadline right now.
We good.
Or you can let me be kind. It's up to you.
I like kind.
All right. So, anyways, if we did a Punnett square, which we went over, right? Remind me, we went over Punnett squares, yes? All right. So if we did a Punnett square and we did two true breeding, we would know that what? What do we know we'd get? We know we'd get heterozygous, 100% heterozygous. We know that today. You know that. because we went over Punnett squares. That means that's one, and what does that mean for phenotypic ratio? It's 100% yellow. And this part here, Mendel did not know. Mendel did this, he just used the Punnett square to kind of look at the numbers. He did not know that about genes, he didn't know about alleles. But we call them genes and alleles today. But he knew that when he mixed these two together, he always ended up with 100% yellow. So he called this dominant. He called this dominant. And he called this recessive. Now they're homozygous. When you see true breeding, anytime you see this key word, they're going to use this a lot. All right, I hope you're listening. They're going to use this a lot in every test you ever take. You see true breeding, you need to think what?
Certain traits.
What? No. I say true breeding. What are you thinking? Homozygous. True breeding. True breeding. All right, so true breeding, homozygous. That's it. Now, did I say dominant?
No.
Did I say recessive? No, I said true breeding means homozygous. So, you have this homozygous dominant, and he called it true breeding. He didn't know, and later he developed it, or we developed it, or whatever, to this idea of homozygous versus heterozygous. So what he found was, when he did this, that these things were independent of each other. They were independent. In other words, the yellow and the green, and the yellow-green versus smooth rough, they didn't go with each other. You could get yellow rough, and you can get green smooth. It's okay. You could get both, all combinations. Now, today we also know that this is another reason why we got lucky, the Mendel got lucky, because even though he had dominant recessive, it is possible for genes to be linked. It's possible for genes to be linked. In other words, linked means, write this down, when genes are linked, they are on the same chromosome. So that means if they're on the same chromosome, then and there was no crossing over, right? And in other words, they're on the same chromosome and they're very close together. Let me try to describe this a little bit to you. So if you have this chromosome and then you have this gene on here, A. If you have another gene on here that's B, okay? Let me use the letters that he used. R and then Y, right? Yellow and rough, or yellow and smooth, right? If they're on the same chromosome, you could get crossing over, couldn't you? Right? So if this is R and this is Y, so they could cross over here, anywhere in this region, right? They could cross over. And that means you can mix them up. That means you can get 4 different possibilities, right? You could get big R, little y, right? We went over that, didn't we? Big R, big Y, little r, big Y, and little r, little y. You get all 4 possibilities because you can cross over here. Does that make sense? Okay. Now, the problem is, and this is again something that Mendel got very lucky for, if these two genes are on the same chromosome and they're close to one another, what that means is that it's unlikely that they'll be crossing over. So if Y were here instead of all the way down there, now there's not going to be a lot of chance of crossing over, because crossing over is random, right? The further they are, the more likely they'll be crossing over in between them. The closer they are, the harder it is for there to be crossing over in between them. So if these two were really close to each other, we would call them linked. So how linked 2 genes are is dependent on whether they're on the same chromosome and how closely they are to one another. Is that clear? Yes. I have one person saying yes. I have no one else saying anything, and I'm hoping that people are getting this. So Mendel got very lucky that these genes were not linked. Because had they been linked, had they been close to one another, he would not have gotten independent assortment. They would not have assorted independently. They would have assorted together. Anytime you got rough, you got yellow. Anytime you got smooth, you got green. They would be together. That does happen with genes. We do have genes that are, you find one trait, you're likely to find the other trait because they're close together on the same chromosome. So this law of independent assortment just means that the characters can assort individually, separately from one another, which means they're not linked, they're not linked strongly. So because of his results, that 9 to 3 to 3 to 1 ratio, remember that 9 to 3 to 3 to 1, that's an important ratio, right? Yes? Mm-hmm. Did I tell you that? Yeah. And we went over it, and I highlighted it. I'm telling you, it's going to be very useful. That's the phenotypic ratio. What we're talking about here is a dihybrid cross, right? We're talking about the yellow and the rough, and the green and the rough, yellow, green, and smooth. Because they independently assort, we have 4 possibilities in heterozygotes, and we have all this 9 to 3 to 3 to 1 ratio that can develop. This would not be the case if they were not independently assorted. So you really need to think about this law of independent assortment as something. The reason I mention this is because we will be going over examples where they are not independently assorted. So Mendel's law of independent assortment depends on the idea that they're not on the same chromosome very close together. All right, so we're going to finish this tomorrow. Try to get this done. We will go over the rest of this chi-square thing. It gets complicated. If you haven't done it, try to read it. And make sure you read that chapter, please. Thank you. Again, this law of independent assortment is not always true, but when, based on what Mendel saw in his experiments, they were true. So now let's take a look at 2 traits, and let's work out the complex, it's not really that complicated, but let's take a look at and try to work out some math. This was something you should have worked on over the weekend. Some kids emailed me and said, look, this is really hard. I said, yeah, it was meant to be hard. You're meant to think on it. Here is a pumpkin fruits with W color, big W, is dominant to the yellow. So let's write it down. What I like to do is I like to write it off in a chart, because when I see it here, right, by the way, this is a clue that you should be doing this too, because you're going to get problems like this on your end of course exam. You're going to get problems like this on your test. This is how you maximize your success. You underline the parts that you know are true. So yellow is little w and white is big W. I like to write it off on the side. Big W is white, and little w is yellow. I like to write it in a chart because then when I can just reference it without having to read the whole paragraph again and confuse myself. And then there's a discoid shape, which is big D, and there's a round shape, there's little d. So big D is discoid, and little d is round. So there's 2 traits, right? 2 alleles each. I know what I can do with that. You know you can do it too, right? You can do a dihybrid cross. By the way, if you didn't do the guinea pig cross yet, pay attention to what we do here, because this is what you're going to have to do. And also refer back to the video.
So when is this due?
Let's talk about that later. As always, you'll have plenty of time, plenty of notice when things are due. So we fertilize a pure plant. I don't like that word pure. You know why? So they're using the word pure. What do they mean by pure plant? They mean homozygous. They mean true breeding. What you'll see in most cases today, we use the words true breeding. True breeding plant, which means homozygous. So let's make sure we understand that. That have yellow discoid fruit. What does that mean? True breeding, homozygous. What do you know about their genotype? So these are yellow discoid. And they're true breeding, which means they're what? Homozygous. What does that mean? What's their genotype? Quick. Somebody quickly tell me their genotype. Is it big or little? Is it big or little? Little W. No. This is the genotype. You need 2 copies of each gene. Both are little W's. Why? Because it's homozygous. And then capital D, capital D. That's the genotype for that. They told me that by telling me what? True breeding. As soon as they told me true breeding, I knew there was going to be 2 of the same. They told me what each color was. They expected me to come up with this on my own. You will be expected to come up with this on your own. All right, we're going to continue this tomorrow. We're going to go through this whole thing in class. I expect you to be working on it on your own so you can come up with what is it I understand, what is it I don't understand, and hopefully ask questions and stay awake in class. And try the guinea pig problem on your own. And I'm going to tell you right now, you're going to run into some problems because most people do. I did when I was your age. I did when I was in college. If you run into it and you're like, wait a minute, how do I make this zygote? How do I cross this? Which letters are going where? That's good because now you're coming up to your challenges. Then tomorrow when you come in, I expect to have people much more motivated and intellectually engaged. Yes? Remember Thursday you come, but then we have half day. The schedule for Thursday is in your Jupiter grades. You should have gone over this in your advisory. Yes? It's parent-teacher conference. Read your stuff in your bulletin. So the genotype for that parent would be little w, capital D. Agreed? And it says it right there. It says pure plant with yellow discoid. Yellow and discoid. Now another pure plant, another true breeding plant that has white and round. So what would white and round be? Big W, and little d. Do you see how you can do that? Assuming you know what true breeding means. True breeding means the same genes. It's real easy. Life is easy. Now you're going to cross them. What does that look like to you? Like the foil. Exactly right. Exactly. You're doing a dihybrid cross. So first thing you didn't know is what is the mom going to give and what's the dad going to give? Well, foil, how many different kinds of children can, or eggs or pollen can this one make? It can only make one kind. What would it be? Little W and big D. That's all their pollen's going to be that, right? And what's the pollen? What's the eggs on this one going to be? Big W, little d. That's the only ones X can make. So all of their children are going to be a combination of little w, big D, combined with, oops. Is it time to go? All right, so we'll finish this tomorrow. Please pay attention. Try to come in and not fall asleep in class, ladies. And big W, little d. So now in the second generation, all the children are going to be the same. They're all going to be. Oh, I shouldn't have done that. Let me erase this. So big W, little d, and little w, big D. That's right. They're all going to be white and discoid, but they're all going to be what genotype? big W, little w, big D, little d. The second generation, they're all going to be this white disc shape, right? Like this, all of them, okay? But their genes are all going to be big W, big D, little d, which means that all of them, when we cross them, now we're going to have a true dihybrid cross, right? Yeah. Now, when we do the next generation, that's the first generation. In the second generation, when we take and we mate these, right? We're going to mate them like we just talked about with the peas. Well, in the second generation, you have big W, big D, little d, cross with big W, big D, little d. What that means is that now you have 4 different gametes, don't you? You have big W, big D, you have big W, little d, you have little w, big D, and then you have little w, little d, right? You have your foil, right? On both sides. So now you have a true dihybrid cross, the second one. So now you get what ratio? I don't have to do it. I'm not going to do it because you should know the ratio. What's going to be 9 to 3 to 3 to 1. If you knew this, if you know that, you know what the results are. All right. That's why I tell you this is such an important ratio. All right. We'll finish this tomorrow. We're finishing this tomorrow. Do the guinea pig activity as soon as you can. Remember foil when you're making the gametes. Please put your chairs up. Let me talk to Santiago and let's move on. All right, so we finished that. We did the dihybrid cross. We looked at the 93 to 3 to 1 ratio, right? And we know that this is a double recessive. Phenotype. And this is where the double dominant. Remember, this is the phenotypic ratio, right? And of course, this is 1 dom and 1 recessive, right? So we understand that that's the results. We saw the chart previously. You can fill it in if you'd like. And also you saw it in the beginning when we did the whole process. And we also did it on Friday. So we've done it twice or 3 times now. Tomorrow you will have to do it for a case study. You're going to have a skin color case study where you're going to have to analyze not a dihybrid cross, not 2 genes, but 3. Not a 16 square chart, but 64 square chart. Yeah?
So 9, 3, 3, 1 ratio always occurs, or is that one only 2 heterozygous?
Those are the kinds of questions that are very, very, very important. So this 9 to 3 to 3 to 1 ratio requires 2 things. Number 1, independent assortment. Which happen, and of course you have to have dominant recessive. Okay? 3, both parents have to be heterozygous. You need to have all 3. In order to get this ratio, these all have to be true. And they'll give you clues in the paragraph. They'll tell you things like, the first generation, they were both homozygous. Both parents were homozygous. After the second generation, once you crossed F2, right, what do you get? Or once you cross the F1s, rather. The parents are homozygous. What happens when you cross the F1s to get the F2s? What do you get? 9 to 3 to 3 to 1. Why? Because both the people being crossed are heterozygous. How do you know that? Because you have 2 homozygous parents. And 100%, 2 homozygous parents of differing genes, right? Just like the example we just did, which means that you have one that can make gametes that are little w, big D, the other one can make a big W, little d. You cross those two, you have 100% big W, little w, big D, little d. So now you have 100% heterozygote. So they give you the clues. If you need to work it out so you understand it in the middle of the problem, go ahead and do it. But the bottom line is they give you clues that lead you down the right path if you understand what's going on. If you don't, well, then you don't. So count the number of purple starchy, purple sweet, yellow starchy, yellow sweet kernels in 5 on the let me see. Can
you go back to the thing?
Here?
Yeah, just working on.
Yeah.
So you know how when you cross two homozygous parents, the ratio is 100% whatever comes out, and how when you cross two heterozygous it's 9 to 3 to 3 to 1.
Yeah.
Is there an expected ratio when you cross a homozygous and a heterozygous?
That's a great question. We're about to do it. It's called a test cross. All right, so let's go ahead and do that. So you're going to look at the yellow, count the number of purple starchy and purple sweet, right? And yellow starchy and yellow sweet kernels in 5 of the rows on your ear of corn, and record the number on your chart. Be sure to use the same 5 rows in each calculation. Make a table and put your data on. Calculate the percentage between the individual traits. All right? Purple sweet, yellow starchy, yellow sweet. The numbers are going to work out the way they're going to work out. Let's go ahead and define. Remember I said what's the first part is making our chart. So let's make our chart. Let's say PS for purple starchy. All right, purple starchy. That's a phenotype. Purple starchy. Let's not use letters because this might get confusing. Purple starchy is going to be what? Let's say that purple starchy is going to look like this. It's going to look. Let me draw this in red. It's going to be purple, but it's going to be full, like smooth. You see that there? That's going to be purple starchy. Are we good with that? All right, so it's going to be smooth and purple. Now let's say purple sweet is going to look like this, all wrinkly, like a raisin. All right? Raisins are sweet, right? All right, so think of like raisins, all right? So purple sweet are going to be wrinkly and purple. Are we good with that so far? Mm-hmm. And with the yellow starchy or yellow. Oh, I think I mixed these up, didn't I? Because we're gonna keep the same. We're gonna keep this the same. So let's keep this the same. Purple starchy are gonna be wrinkly purple, and smooth purple are gonna be the sweet ones. Yellow and sweet are gonna be the smooth and yellow. And yellow starchy are going to be wrinkly and yellow. Everybody okay with that? So let's just have a standard. That's going to be our standard. Now you're going to go ahead and count. And so how would you do that? Let's go ahead and we need 4 rows, so we have to pick 4 rows. So let's pick 1. Oops. Let's pick 1.
5 rows.
Oh, 5 rows, thank you. 2, 3, 1, 2, 3, 4, and 5, right? See, 1, 2, 3, 4. No, that was right. You have to draw this one. So 1, 2, 3, 4, 5. Those are my 5 rows. Okay? Are we all good with that? All right. And it says make a data table. Put your so I'm going to say purple smooth is going to be here, and purple. Purple starchy is going to be here. Purple sweet is here. Yellow starchy here. Huh? Oh my God! Is it time to go?
No.
Oh, what are you talking about? He scared the hell out of me. Don't do that. Yellow, sweet here. Are we good? All right. So let's count then. Let's look for purple sweet. Remember, what's purple sweet? Smooth and purple. Well, let's count the wrinkly ones. I'm going to make the wrinkly ones red. Now let's make them blue. So 1, 2, 3, 4, 5. Do you agree that these are wrinkly?
I thought you started in the row under that window.
Oh, thank you. Let's start there. Very good. That's what I get for rushing this. All right. So let's count it. 1, 2. These are the wrinkly ones. 3, 4, 5, 6, 7. Are we agreed that those are all the wrinkly ones? All right. So then let's use yellow for. Oh no. Let's use red for the smooth ones. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, and 21. So, where's 22?
You just circled the last one that makes it 22.
Oh, okay, sorry. Thank you. So 22 wrinkly ones, right? So purple starchy. Oh, I hate you. All right, so purple sweet, which are that's the red ones, right? So I picked blue. So purple sweet, which are the smooth ones, were 22. Is that correct? All right. Purple starchy were how many? 7. And yellow starchy, how many in that row? So I'm yellow starchy. Let's I don't know, use yellow, I guess. Yellow starchy is 1, 2, 3. Oh, that's a sweet. Sorry, yellow. Okay, let's make yellow sweet then. There's 3.
There's one in between. In between the red and the blue over there.
I don't see it.
Near the middle.
I go the rest starchy, I think. That's how I would call it. Everybody agree?
Yeah.
So I got 3 sweet ones. Yellow sweet, which are the smooth. So yellow sweet is 3. And the last are the starchy ones. Let's go ahead and use, I don't know, black, I guess. So we got 1, 2, 3, 4, 5. Do you agree? Where's the between? You guys keep telling me.
In the middle.
Oh, I see. Where was it? There it is. All right, so how many we got?
6.
6. So there's 6 of these. All right, let's go ahead and do the second one then. Second one, we're going back. We're doing the
Yellow. I mean the blue.
We'll do the blue first. Okay, the blue is the wrinkly one, so that's wrinkly, kind of wrinkly. Agreed?
Yeah.
So I got 4,
Wow.
5. And this is row 1. This is row 2. There's 5. And then red ones. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. Are we okay with that? So there's 24 here. Yellow starchy, let's go with the yellow ones, the sweet ones first. The yellow sweet, so that's 1, 2, 3, 4, 5, 6, 7. 7. And then the last one is the black ones, which are what we got here. 1, 2, 3, 4. Third row. We got, what do we got? Well, we don't have time to finish this, so here's what you're going to do. You have this color version at home in your email. We're not done, so don't put anything away. I still got a couple minutes, so just pay attention. Listen, because you got a lot of homework to do. So listen. You're going to continue this. You're going to continue this chart. You're going to have all five of these. And then you're gonna go ahead then, and you're gonna count the number of purple starchy, yellow starchy, and blah blah blah. And this, be sure you use the same 5 rows for each calculation. Make sure table. You're gonna do it again here. Yes. So this was for this chart and these numbers. You should use the same table for this piece of corn. All right. Pay attention tonight, as far as this lab is concerned, is to continue this and finish counting these kernels. Now, wait for part 2. You're not listening. You're putting things away. You're not writing this down. And so you're going to come in, you're going to say, I didn't know but I didn't know. Now listen. You're counting these up. You're filling in the charts. The second thing you're going to do is you're going to watch a video that I'm going to send you. It's going to be 5 minutes, 3 to 5 minutes. It's going to get you primed for tomorrow's activity. Tomorrow you're going to have until the end of class to complete a Dihybrid Cross on skin color. Now I told you there's a lot of genes involved in skin color, but you're actually going to just handle the skin color as if it were 3 different genes controlling skin color. Okay? All right. I hear talking. This you're not going to do. You're not going to do this. So your job